Hello friends, VBSM Academy, a School of Arts and Science. Today's class is Structures of Carbohydrates from Veerabhandraya Dema. Please contact 9663828428. We will have a brief discussion today is the structures of carbohydrates. Friends, before going to the structures of carbohydrates, what are carbohydrates, how the carbon, hydrogen and oxygen are arranged in the carbohydrates molecules. For example, sucrose C12H22O11 on hydrolysis in the presence of N-vertase, an enzyme gives rise to D-glucose, that is C6H12O6, and the D-fructose C6H12O6. This is the inversion of cane sugar. Here the structures of D-glucose and D-fructose is given for our studies. This is the hexagonal structure of glucose, arranged HOH bonds, and on hydrolysis it gives and splits into its respective components, because sucrose is made up of glucose and fructose. Here, inversion of cane sugar. Come friends, we will have a study of the inversion of cane sugar. This is the alcohol industry. Molasses is a dark colored syrupy liquid left over the crystallization of sugar from the concentrated sugarcane juice. It is about 30% of sucrose, which cannot be separated by crystallization. It is converted into ethanol by the following steps. Dilution. First, molasses is first diluted with the water to bring down the concentration of sugar to about 8 to 10%. Next, addition of ammonium salts. Molasses usually contain enough nitrogenous matter to act as food yeast during fermentation. If the nitrogen content of the molasses is poor, it may be forfeited by the addition of ammonium sulfate or ammonium phosphate. Coming to the addition of yeast, this is very important. Fermentation tanks, we are going to add the yeast is added. The mixture is kept at about 303 kelvins for a few days. During this period, the enzymes synthetase and zymase present in yeast bring about the conversion of sucrose into ethanol. The fermented liquid is technically called wash. In the wash, the important chemical reactions, biochemical reactions will taking place. C12H22O11, which is sucrose plus water molecule in the presence of sucrose invertase enzyme gives rise to the glucose and fructose. Next, this glucose in the presence of zymase, it gives ethyl alcohol, which is also called as ethanol, and liberation of carbon dioxide. Distillation of wash. The fermented liquid containing 15 to 18% alcohols and the rest of the water is now subjected to fractional distillation. The main function drawn into the aqueous solution of ethanol, which contains 95% or 95.5% of ethanol and 4.5% of water. This is called rectified spirit. This mixture is then heated under reflux over quicklime for about 5 to 6 hours and then allowed to stand for 12 hours. On distillation of this mixture, pure alcohol, that is 100% alcohol, is obtained. This is called absolute alcohol. This is all about the industrial chemistry. In the structures are concerned, glucose, fructose, and glucose straight chain form, D-glucose and L-glucose. This is the chain form of D-glucose and L-glucose. D, dextro-rotatory. What is dextro and what is the levorotatory, my dear friends? It is a separate branch of chemistry which is when a plane polarized light is passed through the glucose solution. Suppose the plain polarized light is turns towards right, then it is dextro. If it turns the plain polarized light towards the left, it is levorotatory. Based on the deflection of plane polarized light, the factor is displayed. So these are the sucrose, aura projection of glucose structure, glucose state change structure. This is a keto group. See, this is keto group, and this is as a L-glucose, and this is D-glucose. D-glucose and aldohexose. This is an aldohexose. This is a D-fructoketohexose. This is glucose. OH which molecules are attached here. This is a fructose chain structures. All these structures. Coming to the epimers. What is epimers? Epimers are two monosaccharides that have a different configurations around one carbon atom. You see, this is the carbon atom where oxygen is bonded, that is the keto group. This is hydrogen atom. Similarly here D-galactose. Glucose and mannose are C2 epimers. Number of carbon atoms, aldoses and ketoses. This is the tabular column study. What are the epimers? Aldehyde groups in both the structures C2 and C2. This is the epimers are having CHO group here, CHO group is here, HOH, here HOH. These are the important. This will be discussed later in a separate chemistry. Staple foods of various countries of the world. Staple foods. Day to day, we are using all these staple foods. Starch polysaccharide is a biodegradable natural carbohydrate that acts as a storage of energy in plants and acts as a reserve food supply to the plant. Coming to the photosynthesis. This is plants takes carbon dioxide and water in the presence of light, sunlight. It gives rise to C6H12O6, that is glucose, and gives oxygen. See, plants are a special. This is plants. We have to thankful to the plant kingdom because the carbon dioxide is taken by the plants. That is, the plant kingdom needs carbon dioxide and water in the presence of light, sunlight, and gives what we are breathing, the oxygen. Oxygen is released by the plant kingdom. So, my dear friends, we have to protect the plant kingdom around the world of the planet Earth. Asire usiru tilkodi. Carbohydrates, polysaccharides, starch, glycogen, cellulose are the structures here. So we are already discussed about the polysaccharide examples: chitin, cellulose, glycogen, starch, which are the examples of polysaccharides, which are also carbohydrates, which are also called as carbs. Cellulose fiber, one of the most common types of fiber is cellulose, the main component in plant cell walls. The chemical structure of the cellulose is shown in the figure below with our simplified depiction next to it. We can see that cellulose has long chains of glucose similar to starch, but they are stacked and there are hydrogen bonds linking the stacks. When we eat fiber, it passes through the small intestine intact. Because we don't have digestive enzymes to break it down, then in large intestine, our friendly microbiota, the bacteria that live in our colons, go to work on the fiber. Some fiber can be fermented by those bacteria. What a wonderful digestive system in the human system. So big intestine is having a big factory where cellulose can be digested. This is the structure of cellulose. Observe carefully. So coming to the world of carbohydrates are very important. Starch is storage for plants. Cellulose is a structural polysaccharide for plants. Starch, cellulose, pectins, gums are the important plant origin polysaccharides. Cellulose is the most important structural polysaccharide. As I discussed it today, it is made up of unbranched chains of glucose molecules linked via beta-1,4 glycosidic bonds, already discussed in our previous classes. Well, this is cellulose having its own structure. So this is the size of the cellulose is well depicted in the slide. This is in the meter, this is in centimeter, this is in millimeter, this is micrometer, this is a nanometer, microscopic Armstrong units. This is molecular, and this is the molecular structure of cellulose. Cellulose is the most abundant carbohydrate present in the nature. It is insoluble in water, crystalline solid, having a great powdery appearance, high tensile strength, and due to form hydrogen bonds between the individual chains in cellulose microfibrils. Well, cellulose is the most abundantly, as I told you. Cellulose is also present in the cell wall, other organisms like bacteria and algae. So this is the origin of cellulose, where the plant kingdom is small. This is microscopic. Slowly you can see the things, how it is depicted in the slide. Cellulose, this is the cellulose structure. Thank you. Thank you for listening. This is VVSM Academy, a school of arts and science. Like, share, and subscribe.